Yes, good evening, participants. So, welcome to the 20th session on applications of bioinformatics. That is on virtual screening, genomics, and proteomics. First, we discuss about what is genomics and proteomics. What are the various branches in genomics and proteomics? Then we discuss about how to go for virtual screening using Autodoc Pyrex, PYRX. Okay, before going to today's session, 20th session on applications of genomics and proteomics, um, can I get the last session contents? Can we recap the last session contents? What are the exact topics did we discuss in the last session? You remember I was showing you some hands-on experience with some tool. What is it exactly? Did you practice that? Anyone practice that after who learned new? Or can I ask you a few questions? Yeah, so what are the last session topics? No? So if you interact, hereafter the interaction is most important because the next two sessions will be on mini project discussions, isn't it? So if only if you unmute and answer, it will be best for you. Okay, like if you have any doubts, I can clarify you the doubts. Okay, even you can share your screen. If you have any doubts in the project, I don't know how I can able to bring it up with you because you want to discuss. And if already some participants are doing projects, and even if you have any doubts, I can clarify that. Okay, so the interaction gives you a best information to everyone to reach. Yeah, last session topics. No, what is the software I used for demonstrating molecular talking? Is that I explained in the last session? And whether I discuss something about cellular computing, DNA computing. You remember the topics? Yes, the software I explained is Autotalk. Very good. So okay, we had a gap. Now only I came to know. Yeah, Saturday, Sunday we have a gap. Okay, so you forgot. But continuously I'm reading something and I'm doing something. So I forgot about all this. What I mean, why you are lagging. Now I can able to get. Sir, can you explain about estimated inhibition constant in the .dlg file? Should we also need to consider estimated inhibition constant value along with the docking score to check whether the compound acts as a receptor inhibitor? Actually, the dock score is what we call it as negative binding energy score. Okay. So whatever we completed on last session, okay, I think this is the demo. As you have mentioned, this is .dlg file. So doc1.dlg file, I show you in large icon, so you understand. So we have, as you have mentioned, this is the doc1.dlg. You can open it in Notepad software or even WordPad. As you mentioned, just because I just tried with only two courses, we have received two scores. Okay, two different post scores. I can show you the score and I can explain. This is for model one. I can increase the zoom it and show it to you. You remember we have used genetic algorithm. We have used genetic algorithm for docking. Just slightly reduce. Yeah. So if you see this estimated binding energy is minus 6.56. Okay. And how you got this? So of course, this is a summation of all the energies. You have energy like also final molecular energy, van der Waals hydrogen bond dissolution energy, electrostatic energy, final total energy. But the most dominant energy, what we take it for granted, is this estimated free energy of binding, which we got as minus 6.56. This is for post one. Okay, we also have pose 2. I believe last time I showed you minus 8 point something. Okay, for model 2, you got more negative maximum. If it is minus 6 and below, you tell it as a significant energy. So now we have minus 6.56, so it is significant. But if you see the model 2, okay, if you see the model 2, as I already mentioned, you got like minus 8.80. It's more significant when compared to the previous model. And according to your question, estimated inhibition constant is dependent on the binding energy. Is dependent on the binding energy. Here we have minus 8.8. So inhibition constant, what we got is 357.05 nanomolar. Okay. So generally, if you want to go for estimation of drug dosage, okay, like the drug here we mean as it is used as an inhibitor, isn't it? So in pharma, we call it as agonist, isn't it? So the drug has to bind in the receptor, and the kind of inhibition we constant we can measure the dosage. But whatever we identified, it can be limited up to the in vitro. So one has to go even with in vivo. Then the experiments has to be conducted, and even the dose has to be found out accordingly. This is something like I show you 357, isn't it? If and if I try with three poses, you can have a idea on that. So for the negative maximum, you have more. You have less dose when compared to this. How I am telling that is less because that is mentioned in nanomolar, but here it is mentioned in micromolar. If binding energy is more, you can reduce the dose of the drug. So if it is 15.56 micromolar, it means that if you take it in nanomolar, uh, 0.01556, isn't it? Okay, is that? No, 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 no. 15.56 micromolar is 15.56 into 10 power 3 into 10 power 3 nanomolar. Okay, so you are taking more drug here. Okay, more drug. So through the binding energy, the estimated inhibition constant is calculated. Of course, there is a correlation for it. Okay, it's directly proportional. Even if you do multiple docking, check with whatever ligand and receptors. So one has to find that only through the autodoc literatures. Sir, please, is that clarified, Crasto Tony? I just mentioned that in the DLG file, we have estimated inhibition constant and docking score. Based on the docking score, that is, if you have a negative maximum score, then the inhibition constant value will be minimum. Okay, that's what we anticipate. If you can able to take a low drug, and if it can able to cure, if it can inhibit, that's what we want, isn't it? Is this okay, Crasto Tony? Do you want further explanation? Good, thank you. So this kind of interaction is wanted. Next, sir, please explain how to get all the residue interactions in Excel. How to get all the residue interactions? Okay, in Excel, isn't it? In Excel. Mm, that's not possible. But one can try with what if. So in what if, whatever PDB file we have created. So you okay. You want it in Excel, isn't it? Now I got it. So we can able to create. Okay. So as such, we can't able to generate in Excel, but we can able to create in Excel. Okay. You want some kind of a solution for that. Now for this, I opened the complex. That is, you remember on yesterday, I mean last session, I just saved the ligand and protein dot underscore complex dot pdb. Okay, this is the file. I'm going to open it in Discovery Studio. In Discovery Studio, we can see 3D interaction and 2D interaction. So this is the 3D interaction. I just uh, gone to the display style by clicking right, and then graphics. Instead of background color black, I kept like white. Okay, it looks elegant if you see it in white background. However, you find this, if I click show T2E diagram, you get like this 2D diagram. But sir is asking like how can we bring the interactions? Okay, to know the interactions, I also give one more alternate choice. Like we have to click view. Okay, here we have to keep view hierarchy. So what I made is I click view hierarchy. Okay, and then I'm going to go for the next step. Scripts selection. Select ligands. So it has to select only the ligand alone. Whether it's the yes, the ligand is selected. So only you're getting yellow color. Previously we had a multicolor. Now the ligand is selected. So the step what I did is script selection select ligands. The next step is select atoms within 4 angstrom. So near the 4 angstrom, you can find the binding sites. Okay, whatever atoms present near to the ligand that is close to the 4 angstrom, it shows the result. So for that, I have completed that step. Second step. The third step is ligand interactions. Show ligand binding site atoms. Ligand atoms. Ligand interactions. Show ligand binding site atoms. Now, this is the hydrogen bond monitor. You click here and you find here. Okay, so this is the data. Okay, whether I can able to zoom in. I think zoom in is not possible. So I can able to show up to this. So that is, if you see the ligand, okay, so this is the ligand MK1902. MK1902 is interacting with glutamine 2. And similarly, if you see here, this is one interaction. Okay, the next interaction, okay, the next interaction is this. The third interaction, this is a ligand-ligand interaction. This can't be taken. So this is the second interaction. Actually, two binding site amino acids we have. We got like glutamine 2 and asparagine 98. Only two binding site amino acids are there. So this we have to jot down in a form of a table. Okay, like how we can jot down in the form of a table. If I take a Microsoft Word, okay, so I can uh, give you a tip like insert a table. Okay, just zooming in, or even the layout I can keep it in uh, landscape. Okay, check it out. Still not maximum. I fit distribute auto fit to the window. Okay, this is font. Can I increase to 28? Yes. Now, so with the data, whatever I have, first amino acid uh, position and atom position, or here itself amino acid and its position, and atom binding atom position. So we can create a table. So only we can create a table like this. Okay, because as such we don't have TSV files or generated. So this is up to the receptor, amino acid position and atom position of the receptor. And if you want the ligand, okay, the ligand atom and ligand position, okay. And if you are getting an internal binding energy score, internal binding energy score, which we generally represent in kilocalorie per mole. In Autodoc, we get like kilocalorie per mole. So we got two binding sites, okay, for this glutamine and atom position. If you see, no, glutamine 2, the atom position is He22. Okay, hydrogen epsilon 22. And then In the ligand atom, O and this is not, this is O, oxygen and position, you can mention like 3. Okay, so for what I opened, if it is a second pose, which we got -8, you can give this -8. Now go to the second, you can give this aspersion. So we have to manually create. Okay. So after the hierarchy, you can follow this and you can manually create. So whatever I am showing here or the hydrogen bond interaction that you have to remember. Whatever I showed, okay, or the hydrogen bond interactions. And if you see here, if I maximize it, okay, other than hydrogen bond interactions, we also have van der Waals, conventional hydrogen. Whatever I told glutamine 2 asparagine, the green lines or conventional hydrogen bonds, but we also have a carbon hydrogen pi donor. The pink is pi pi, okay, and the dark pink is actually pi pi T shape. So we have different kind of interactions here, isn't it? So if you want to represent, even you can represent it, and if you want to change the display style here also, we can change it. Okay, for example, in atom we get a line one, but if you feel comfortable with the stick model, you can make it in stick color. If you want to, in the element, so you can create like this. You got it. Whatever you find in the 3D interaction, similarly, I'm bringing it in this 2D. Okay, and residue color by interaction. And if you want this residues in the form of disk model, so see how a disk looks like. Okay, the color is elegant, isn't it? So an interaction, if you want a bond distance, show distance. So the bond distance is shown now. So these are the additional data I can provide. If you see here, earlier we don't have any bond distance, isn't it? So glutamine 2, I can use 3.64. So this you can give as an additional thing. So binding energy, you keep it in the last column. Okay, so we can keep it in the last column. Before that, what kind of bond is this? Category of bond. Uh, Category of bond is the conventional hydrogen bond, isn't it? Give like conventional hydrogen bond, whatever. So, and here you will be giving like bond length. So whatever bond length you got, okay. For example, here you got like how much? 3.64. For this 3.64. So likewise, you have to frame a Excel sheet, or you can use a Word to jot down the values from whatever you got from the Discovery Studio. That is after docking. Okay. So this is how we can tabulate the values, and even you can see the look and feel effect of this 2D is fine, isn't it? So you just keep close to that so that you can minimize the area, and now you can take a snapshot. Okay. What exactly is the next question? Uh, this is okay, sir. Dr. Maheshwar. Yes, can you please also explain docking validation? How to validate docking? Yes, good. Again, nice question from your side. Actually, the docking, what I tried docking the drug called Intina with protease, HIV protease. Okay, if I know that protease, I'll go to the protease and just find whether it's modeled or how this is done. This is a small one, I know. I don't think it's a kind of a model. If it is downloaded from a PDB website, for example, if I take you to the website called PDB Sum, there is website called PDB Sum. Okay, if you go to this PDB Sum, to the other participants, exactly what is this question is asking like if after the docking, I am mentioning like asparagine 2 and glutamine 91, isn't it? These two are the binding sites. But how come I can tell that we can tell that these two amino acids are the binding sites of that enzymatic protein protease? How we validate it? We are getting binding results. We are getting hydrogen bonds. We are getting other bonds also. Okay, but how to validate that? For that, we have to go to the appropriate some tools for checking whether the binding sites are correct or not. Okay. If I have used one kind of ID like 1KFP, in that case, earlier whatever I tried is a modeled receptor. If so, I am not getting any PDB coordinates. Okay, at the top itself, I will be getting what is the PDB ID and what is the header file information, all this information. But I don't have anything. So I try with this. Okay, a sample file, whatever given, or I can try whatever we have. For example, if I want to try with spike like a protein, it takes more time. Okay, no problem. I can take it. So I want to try with a spike. I mean binding sites for the spike glycoprotein. So I'm going to give like spike or SARS-CoV-2, whatever it is, spike glycoprotein. I'm taking you to that. So now I am finding for SARS-CoV-2, 1830 results are there at present. So these are some of the domains. Okay. If I want to find for this one, okay, actually, this is spike glycoprotein already complexed with the human receptor ACE2. You know, the spike glycoprotein, if this is the spike glycoprotein in green color, this is interacting with human ACE2 receptor. So whatever binding sites, whatever amino acids of SARS-CoV-2 spike contacting with human, that is angiotensin converting enzyme. Okay, the contacting amino acids is what we call it as binding sites. If I try to dock my drug, okay, with spike glycoprotein, and if the binding sites of ACA2, which is binding with, you know, how many binding sites will be exactly binding with hydrogen bonds? 13 amino acids out of 1330, 1333 amino acids. Only 13 amino acids are contacting with each other. If your drug is going to dock in any of the 13 binding sites, okay, even if it docks nearly 3 to 4 binding sites, your drug is a valid drug, is a valid inhibitor for spike protein. Okay, so that's a kind of inference. So for this, okay, I just getting inside 6L is at G. Okay, so you have to type here 6L is at G. Fine. This is an example. Generally, if you can't find such a kind of example, the SARS-CoV-2, I think many know that. But if few people don't know, generally, this is how we have to understand that a viral protein generally interact with human protein. Then what is a human protein? How it interacts? What are the binding site amino acids? So for this, I'm going to show it using PDB sum, which is present in EBI portal. So here you have protein-protein interactions. We have one entry. Before that, I just drag and show you what exactly we have. So we have a description of that, and we also have an angiotensin converting enzyme from Homo sapiens. Okay, the size of the protein is mentioned, and this is generally denoted by chain A, and chain B is from the virus. Okay, so whatever we have, if you see the chain A is 596, but the spike protein they are mentioned as 195. It means that they are not docking exactly with the spike protein, but they're docking with receptor binding domain. This is most important. The spike receptor is 1333 amino acid, but the spike receptor domain is limited to 195 amino acids. You know, it comes around 310 amino acid to 500 something. It comes like that, okay? So 300 to 500 amino acids of 1333 amino acid is the binding site domain. So I told you the exact binding sites are 13, but even 195 amino acids, if you find your ligand or peptide is going to dock, it is going to have some effects, but not a best effect. The best effect, if you want to get, that is, if your inhibitor is best docking with the spike, I mention you to go with 13. So where can I find that 13? This For this, click protein-protein interactions. See here. So chain A, they have mentioned like human AC2. Okay, you have this much binding sites. And if you see chain B, this is the spike glycoprotein domain. That is part of the spike glycoprotein. So how many amino acids shown here? These are the binding sites. Okay. And even in the spike glycoprotein mutations happen, alpha, beta, gamma, delta. So many mutations happened, isn't it? So if you have a raw SARS-CoV-2, I mean the initial SARS-CoV-2 in 2019, we have 13 binding sites. But here you have nearly 11 hydrogen bonds. Can be a mutated protein, but you have 11 hydrogen bonds, one salt bridge. Okay. And number of non-bonded is 115. Of course, it's more. Okay, 11 plus 1, you have a kind of a base bond. So almost 12. Okay, so 11, 1. You can find a kind of a bonding with chain E and chain B. So this is one such binding sites validating website. Other than this, we can also find the information through UniProt. You remember? So I already mentioned you that this database is used for fully annotation database. If you want to get more information of a protein, better go try to explore using UniProt. Now I am typing UniProt spike glycoprotein. Default is as spike SARS-2 is there. Get inside the entry, the first link, and now so one can go to search for binding site domains by clicking. We have so many parameters here, isn't it? Can we go to the interaction? I'm just clicking the interaction. Okay. Now we can find some information. So binary interactions are shown. Okay. Even this kind of this is what we are getting some information of the number of experiments and kind of interaction. Okay. And now I'm tracking below. So you can find some kind of interactions information. Protein interaction, we can also check with BioGrid. Okay, and also, so this is some information, but even in top, we can find some binding site amino acids. Yeah, now see here, uh, we have different positions. Okay, we have different positions, and even the motifs are mentioned here. Motifs are mentioned. So what is most important binding site amino acid? So this is what 319 to 541. Okay, here only the AC2 is going to bind. So even the binding sites can be determined through UniProt. This is an evidence. If publication is there, you can find it from the publication. Got it? So this is what they have shown around 195. Huh? So, but if some kind of numbers are not matching, then it might be a kind of a mutated, no doubt in that. But even if you have numerous spike glycoprotein mutations, we have only one entry in UniProt. The one entry what they're showing is the wild spike glycoprotein without any mutations. Even mutation information is also available in UniProt. If you drag and if you see that, you find some data on that. Okay, so this is something about um, how to identify binding sites. Okay, anything else? I think I have explained, Dr. Mahesh. Is it so? Is it clear? Still, if we can't able to find binding sites for your protein, I suggest you to go with CASP-P. So there is a software called CASP-P, which is Computed Atlas of Surface Topography of Proteins. So here you have to feed the PDB ID or upload the PDB structure what you have modeled. So if you have even a modeled one, we can able to upload it or else just we have to feed the PDB ID. You know, so this server is going to determine us various binding sites, that is hydrophobic and hydrophilic binding sites are clearly shown here. So we may have multiple binding sites. For example, so each and every binding site is shown in the form of a sphere. We have multiple spheres here. So almost based on the binding sites, we have that much kind of a spheres here. So for our protein, if you type the PDB and check, you can find similar kind of binding sites. Okay, this will just tell you whether if it is a 319 to 541 is the binding site, whether it's a hydrophilic site or hydrophobic site. Okay, of course you will be finding that as one of the site, but what type of site? All these things you can find from the cast field. Okay, easier. Just type the PDB ID and search. Can you try it? 6LZG. 6LZG. Search. What happened? It's getting loaded. It takes some time. Cannot find. What happened? Is there, isn't it? 6LZG. Mm, then can we go for uploading? If you have downloaded, you can upload it, but no multiple NMR structures. Only one structure can be uploaded at a time. Okay. So one can download this and you can upload it. You will be getting an answer, no doubt in that. PDB format we have to download. So I drop it in demo. I'm going to upload here in Bioinformatics demo. And any other doubts? Meanwhile, the others can also think and ask me. If you have any other doubts, link with it. Email is an optional. Simply I'm clicking submit. Let us see what's going to happen. I submitted. Okay. You'll be getting results instantly. More out of that. Take some time. Okay. Any other doubts? Can we report all this in the paper? Will it be considered as a valid source? Yes. Whatever I showed in Uniprot, whatever I showed in PDB sum, it's a valid kind of explanation for the binding sites. One can able to show this as a reference. Even if we have references parallelly in Uniprot, you can show that as the major reference. Okay, many references are already cited accordingly. Okay, now then I think I can go ahead with the present topics on genomics, proteomics, and if time is there, I can explain about virtual screening or we can postpone it on tomorrow. Now, genomics, 1000 genomes project. For if you want to go for genome, how can we go for a human genome? Multiple windows are open. Can close. Okay, how to open a human genome? If you want to see it in any human genome, okay, we can go to genome, NCPI genome, and you type the species name. Okay, so generally we search like this only. Okay, genome you can give like Homo sapiens default. It's getting selected. No, no, no. I can increase the font. Come back. Come back. No, I'm going to genome. Homo sapiens genome is different. Homo sapiens mitochondrion, you get a different genome. In Homo sapiens genome, we have 22 haploid autosomes, X and Y. So totally 24 chromosomes map this human genome, which is circular in shape. And mitochondrion alone is shown as a separate genome. So if you want to go for checking that 24 chromosomes genome, okay, I'm just selected like this. We are getting various entries for this, for all the chromosomes, and we also it linked with the genes. Okay, so this is how we can take it. So if I want to go to an assembly, I am clicking assembly. It takes you to this. Okay, so for the assembly, you have an ID, you get some information. If you want to see the same, Uh, you see this? This is the size of the human genome. This is the size of human genome. 24 chromosomes are taken in consideration. How many organelles? Only one organelle. What is organelle? Nucleus, isn't it? So only the nucleus can able to find the chromosomes. But in mitochondria, which is a different organelle, you find there also can find only mitochondrial chromosome, only one. So we also have contigs, scaffolds. Okay, these are the terminologies we use for the fragments of DNA. I mean, the fragments of DNA sequences of chromosomes, we call it as contigs. If you map 2 or 3 contigs, you get a scaffold. The scaffold is a combination of few contigs. Okay, so we also find this information like how many is going to code, non-code, so and how much duplicated, fragmented, missed, all this information. If you want to go to a specific chromosome, for example, if you want to go for a chromosome 11, click here. It take you to the specific chromosome. And also we can have plenty of information for the that is if you want to find more details, either clicking chromosome 11, it takes you to the same, or even going clicking here, it takes you to the same kind of thing. Okay, if you want to see the genome, we can able to see the genome using UCSC genome viewer. Okay, UCSC genome viewer is generally used for viewing the genome. Now I'm clicking this chromosome 11. You know, the chromosome 11, we find insulin protein. So just I click the chromosome 11. The chromosome 11 is displayed at present. Okay. We can find short arm. We can find long arm. If you want to specifically check the insulin, okay, this is a UCSC genome viewer from NCBA. So from genome, I am taking you to the genome viewer. We are in chromosome 11. If you want to check with other chromosomes, if you click any chromosome, it takes you to that chromosome. We have information to track blast. If you have a small part of a sequence like insulin gene, if you want to blast your insulin gene and want to find the loci, okay, where the insulin gene is present in the chromosomes, you can able to find it. Okay, like how you use a blast. Here, the genome blast, we use a BLAT tool. And if you see this genome, that is a chromosome, okay, the chromosome, you can find NCBA reference sequence annotations, genes information. This is RNA sequence exon coverage, RNA sequence intron spanning. So these are the various spanning regions of the chromosome. Okay, so these are the, it takes you to the genes. Okay, you can also select a particular as a set of gene information and you can find in detail. If you explore, you can zoom in and you can find the details. Okay, so if this is about UCSC genome data viewer in NCBI, okay, we also have a BLAT tool from Ensemble. There is one website called Ensemble. Inside the Ensemble, so one can go to the BLAT tool. One can go to the BLAT tool, paste your sequence, and you can try to find where exactly that sequence is present in genome, human genome. Okay. For example, if I have, if I'm going to take insulin gene from NCBI, okay, I'm taking a different window. NCBI, get inside the NCBI. So ncbi.nlm.nih.gov. I'm taking you to the nucleotide or gene. Okay, now I'm going to type something like insulin. So whatever sequence I'm going to get for insulin, for Homo sapiens. So you just click here to filter it. Okay, I'm going to get a nucleotide sequence for a human insulin. So if this is your complete CDS, complementary determining site, around 4,969 base pairs. So I'm not taking the protein, but I'm taking the DNA sequence, and this is the size. So one can able to get the FASTA format, copy the entire format, okay, and then whether it's still asking FASTA format, paste the FASTA format, okay, and you can search for. You are using a search tool, BLAT, okay, a DNA sequence default genomic sequence, okay, a description if you want to give, we can give. If you want to give even email ID, you can give, or simply click run. So I just pasted an insulin full, you can see this is an insulin complementary determining site, that is insulin gene. I just pasted in the BLAT tool, that is which is present in ensemble, and checking with the aligned regions in human genome. That is, you're taking a nucleotide, searching in human genome. So it's happening here in ensemble. The same, we can also try the same here also. We have also here blast tool. So one can also try to use this tool. Okay, click new blast and try giving an input for that. So now it's going to search which chromosome and where exactly you can find that kind of a sequence. That is 4,900 character sequence. Okay, likewise, an unknown error occurred. An unknown error. Okay, we are getting like this. So, but just before I tried, I also got some information. I close whatever you already learned. Can we try the sequence here? No, I'm getting something else. See, other than FASTA format, I'm getting few more information. So only I'm not getting this info. So I'm not getting the result. If I pasted, I found this is not a FASTA format. Okay, bottom I got some information, unwanted information. Now this is right. So you know this is from genome.ucsc.edu. This is from different website. In NCBI we have this genome viewer, but also in UCSC also we have this kind of a genome viewer. But initially I'm going to use the BLAT tool, searching on a gene in the genome. So after pasting that insulin gene, I am feeling lucky. This is what you have to click. Similarly, here, okay, I think I have some unwanted sequence, isn't it? I don't know why it's showing like this. Or I can take you to the website where I just tried. Oh, this is what we got. I just searched the insulin gene, you know, I got this result, chromosome 11. We want to come back and show once again. So this is the website. I have used multiple windows. So you're so near me, and you know, in assembly, I'm defaultly using the 2022 assembly. You know, the first human genome was drafted and published in July 2003. So the recent genome posted in January 2022. So that I have selected. Okay, and now I'm clicking. I'm feeling lucky. If you click this, okay, the insulin gene location is identified here. So if you drag it, okay, it's in this position. It tells you what position, size, etc. Okay. And if you see here, you get also custom tracks, configure. You want to go to a particular location and find various informations. So one can also go to this. Okay. So there are plenty of ways to explore about the genome. Okay, if this is about a single genome, if you want to go for 1,000 genomes, we have a different URL for that 1,000 genome project. If you click 1,000 genome project, it takes you to the internationalgenome.org. Okay, like if you're working on a cancer diseases, I suggest you to go with 1,000 genomes. If you are searching a cancer target protein, okay, and like a cancer target protein can also vary from each and every patient. May a target protein in a patient, you may find one or two mutations, which can give us a different kind of information. Okay, so if you get a different information or not, that you can check with 1,000 genomes instead of checking with a single genome, like how I tried now using UCSC genome here. Okay, so you get more precise information if you go with multiple genomes. This is established in 2015. But if you want to find precise information for rare disease syndromes, that is, if one in million is going to suffer from a disease, if you want to find an accurate information, instead of restricting up to the 1,000 genomes, we can go with 1 lakh genome project. This 1 lakh genome project was initiated by Genomics England and completed in 2018. In 2018, we have 1 lakh human genome project. Not only in human, but there are also, we have projects for well-known species like Oryza sativa, rice. I think even for mice, I don't know, we have to check, but we have for human the plant like Oryza sativa. So this is most important, isn't it? If you go for the rare disorder syndromes, one can go to this so that it can get precise information. Okay, so this is something about whatever I mentioned there. Okay, I take it to the website and may explain. Generally, if you want to treat cystic fibrosis, Huntington disease, or even the diabetes, heart disease, for this we could generally use 1,000 genome. If you see the 1 lakh genome project, which is completed by Genomics England, actually completed in 2019, not 2018. And they announced this as a data release 7, which included the 1 lakh old genome. This is in June 2020, the Lifebit established this 1 lakh genome project publicly free for the academic research institutions. Epigenome, if you are concerned with methylation, if you're, I think this I have already explained, isn't it? Epigenome, pangenome. You remember the difference between epigenome and pangenome? So even some diseases are caused because of post-translational modifications. So the best example is cancer, which is arising because of PTM. Okay, the dominant one is a DNA methylation. Okay, so if you want to find a kind of genomic information based on the mutations happened because of PTM, one can go with epigenome. Okay, so this is helpful for you. And pangenome is generally, this genome is sequenced from 47 species. I mean, 47 different ethnicities in human. We have a human pangenome which is established from 47 ethnicity. I told you an example like Asian American is different, Latin American is different, Native American is different. So for each and every type of citizen, okay, we have a genome, including Indian, okay, we have a genome. So all these 47 genomes together, they had a kind of assembled genome called pangenome, which we got it. This is a set of 47 genomes, of course. We have for other species also. This can give you uh, to use a limited genome, okay, instead of you going for 1,000 or 1 lakh genomes. This will be helpful for treatment of few diseases. And pangenome versus core genome, comparative genomics. Okay, I told you what is core genome, pangenome. Okay, now comparative genomics. So if you want to compare uh, the human genome with other species, or closely, generally go with closely ancestral species, isn't it? So if you see, the published chimpanzee genome differs from that of human genome by just 1.23 percentage in direct sequence comparison. So only we are telling chimpanzee is the immediate ancestor of Homo sapiens. Okay, and the other species like gorilla, orangutan, there is much. There is no doubt in that. One can able to conclude only through comparative genomics. That is, you know the size of the human genome, which is 3.19, 3.1 gigabase pairs, and whatever the size of the chimpanzee, I think chimpanzee size is larger, 3.2 something like that. If only if you compare all these genes of chimpanzee and human, you can come to know how much similar you have. So you have only this much deviations. It means that 99.77, 98.77 percentage or similar. So also given some examples, see, a major difference between the two genomes is human chromosome 2, an example they are providing, which is equivalent to a fusion product of chimpanzee chromosomes 12 and 13. You know, the 12 is considered as equivalent to a chromosome 2A, and the chromosome 13 is considered as chromosome 2B. And this is a genome data viewer, how it looks like. And generally, we have a genomic DNA, which we can uh, use a shortcut. We can split into multiple fragments. Okay, whatever we create an artificial library, if it is from a bacterial source, this is a bacterial artificial chromosome library. We organize it, map large clone context, whatever we map, okay, we find a bacterial artificial chromosome to be sequenced. So the again you use a shotgun, and whatever shotgun we use, go for mapping, and after mapping we get a sequence assembly. Through the sequence assembler, the final assembled sequence can be retrieved. So if you want to go with a genome that is taking the chromosomes from the nucleus and start sequencing, okay, generally we follow these steps. Okay, we start with the clone context and come up with the end up with a sequence. There are different sequencing technologies, isn't it? Celera, Illumina. Okay, so majorly you have to remember whether we are mapping the whole genome or we are mapping a particular chromosomes. Okay, that is a say entire set of genes in the chromosome. So that matters. Okay. Here genome sequencing, one has to remember that genome assembly is of course an important step in genome sequencing. Genome annotation. You know, genome annotation consists of three main steps: identifying portions of the genome that do not code for proteins. Number two, identify elements on the genome, a process called gene prediction. I believe that I told you how to go for gene prediction. Okay, so use that is also a part of a genome annotation. Identify a portion of the genome that do not code for proteins. So I have taken you to the human genome. Okay, in bottom also you find something 98%. If you click that, it takes you to the non-coding genes. Okay, and attaching biological information to these elements. Okay, so this is also already there in the genome. You know, one of my I am a DC member of one of a PhD scholar who is doing her PhD research in sequencing a plant genome. Okay, nowadays sequencing a genome is like a PhD research is a favorable PhD research topic if the genome is not yet published. So she is sequencing a novel plant genome. No one has tried that. So if she has successfully complete that, she has to go for annotation. She has to consider all these points, whatever I mentioned. She should have the structural annotation. She should have ORF and the localization, gene structure, coding regions, location of regulatory motifs, etc. If it is functional annotation, We have to find this information, biochemical, biological. Got it. So these are the information generally we needed other than the genes information or mapped from the clone context. Okay. So the annotation is also important for genome. So genome plat, this is one of the tools to find exactly where your DNA sequence, especially cDNA sequence. From RNA, if you want to convert to cDNA, if you use reverse transcription, so exactly you are using a single stranded mRNA, which is equivalent to cDNA. If you want to search, we can search where exactly you can find in a chromosome using this genome plat. Yeah, in genome plat, we can also see not only gene-gene interactions, also protein-protein interactions. We also have an interaction networks here. Okay, for proteomics. Then what is proteomics? Proteomics is a large-scale study of proteins. Proteins are vital molecules. Uh, if you can also have a separate human proteome. Okay, then what you call proteome is the entire set of proteins produced or modified by an organism or system. So the proteome can be species specific, or even if you cluster few related proteins. For example, if I want globin related proteins in the form of a proteome, I can able to construct. That is actually what you do. You create in the form of protein clusters, hemoglobin, carboxyhemoglobin, myoglobin. All these things also you try to construct in the form of a proteome. Actually, how the proteomes are, there is no such shape for a proteome. Generally, we use mass spectrometry techniques to identify the proteins based on molecular weight. And if it is 2D gel electrophoresis, we are estimating, in addition to the molecular weight, we are estimating the isoelectric point. Okay, so whatever I mentioned, okay, so this is pertaining to the proteomic study. Only to the wet lab studies, okay, whatever you generate the data, we generate the data from the mass spectrometry. Through the computational, we get this kind of an information. Okay, and if you see the proteomics, two types of proteomics are there: top-down proteomics, bottom-up proteomics. A slight difference, okay, in between this and that. Okay, if you take a biological sample, extract a protein, if you separate and quantitate proteins using the gel electrophoresis, okay, this is a kind of top-down approach where in the bands you can find the proteins in the form of a bands. But whereas if you take this cell and you go for enzymatic, after protein extraction, you go for enzymatic digestion, and after the digestion, if you start sequencing, then this is bottom-up proteomics approach. Two types of approach is there, top-down and bottom-up. This extract I have taken from Wikipedia. Proteomics software, we have plenty of softwares. Whatever output generated from mass spectrometry, TOF, okay, MALDI TOF. So all these things we can able to predict from the proteomics software. So of course, even peptide mass fingerprinting, a part of thing we can able to find from. Uh, and if you want to see the proteomics software, plenty of software is shown in Wikipedia itself. In database search algorithms, you have so many softwares, and de novo sequencing algorithms. We have MS peptide quantification, which is a part of a protein. We can find this Wikipedia information. So I believe I have showed you sufficient information on genome, proteome. The virtual screening is kept pending. Anyhow, tomorrow I'll be taking you to the first how to work on virtual screening. Then I have planned to project on this mini projects, explanation of this mini projects, how to take. Okay, I can explain you with a kind of a case study also, if possible. Okay, so we have six topics to discuss. Actually, genome annotation function analysis is what we are discussing it on today. Protein structure validation, I think you know, but how you can take it in the form of a project. how you validate, all these things. If anything is uncovered, we can discuss phylogenetic analysis. Okay, what are the various tools and how we can bring in the form of a mini project that I will be discussing, and even drug discovery and virtual screening, network analysis, system biology, that is especially cytoscape and how the MATLAB tools are used. Okay, and immunoinformatics approach. This is something new which is uncovered in bioinformatics, immunoinformatics, dealing about immunological molecules and peptide-based vaccine design. Okay, some of the titles we can discuss this on tomorrow. Can we report all this in the paper? Will it be considered as a valid source? So this is old comment. Okay, any other doubts do you have? Any other doubts? If no doubts, thank you very much for your patience. Even if you're working on some other projects, if you have a doubts for clarification, you are welcome. Okay, I give a chance for presenting your PPT or your desktop. You can project it, and if it is possible, I can clarify. Okay, or I can go with my presentation contents. That is what is uncovered that I will be teaching you, which is useful for executing the projects. Okay, just 2 sessions tomorrow and day after tomorrow. Don't miss these sessions. You feel more important. Okay. So, if no doubts, as usual, uh, we can meet you on tomorrow. Nice day to you all. Bye.